Okay, thank you again for the invitation. And what I'm going to talk to you about, I think it's just was briefly mentioned, is mostly our studies on the role of satellite cells in muscle hypertrophy. And for most of the talk, what I'm going to talk about, the model that we use for studying muscle hypertrophy is synergist ablation, which I'm sure most of you are aware of, and that is where we remove a small portion of the gastrocnemius and soleus muscle, most of the tendon, such that the plantaris muscle then it hypertrophies tremendously. And this is the model that we use, and details of this surgery. We published detailed methods paper several years ago. So I'm going to just briefly give a little bit of background of work that's published. And the mouse that we used, we started studying the role of satellite cells in muscle hypertrophy, was actually using the PAX7 DTA mouse, that is the CRE-LOX technology, where PAX7 drives CRE and thereby activates the diphtheria toxin A gene, so it effectively kills satellite cells. And it's very effective, and one can deplete more than 90% of satellite cells. And with then subjecting the animals to the synergist ablation surgery, what you can see is that normally you do see nice myonuclear accretion with synergist ablation, which is completely ablated with the tamoxifen treatment of the mice to deplete the satellite cell pool. And what we showed is that actually the initial phases of hypertrophy for the first 2 weeks are unaffected. So the muscles grow in the first 2 weeks. However, over time, the growth in muscle weight and by fiber cross-sectional area is in fact blunted. And looking at the muscles here after 8 weeks of synergist ablation, although it is possible, and we're still trying to determine if there is really a myonuclear domain ceiling, that is that the muscles can't grow because they need another nucleus in order to ramp up transcription and translation. But what we have begun to study in more detail is that what we think is really important in this blunting of growth is that the muscles become fibrotic. And that's shown here, both by picroserous red staining at the 8-week time point. You can see that in the absence of satellite cells, that is following tamoxifen treatment, the muscles become collagen abundance is much increased, as is glycosaminoglycans identified by WGA staining, wheat germ agglutinin staining. You can see that even here quantified below the WGA staining is that just even in the sham operated animals, you see an increase in the abundance of extracellular matrix surrounding muscle fibers, which is greatly increased with this overload. So that it really appears that potentially the muscles are just become embedded in this extracellular matrix, and we think that this fibrosis is actually impairing growth. And the mechanism at least one of the mechanisms contributing to this, I'm going to summarize just in this graphical abstract because this work is published, is that through a variety of experiments, both in vivo and in vitro, co-culture experiments, and a number of other studies that we performed, published 4 years ago, that what we believe is happening is that with, in this case, synergist ablation, you have an activation of satellite cells, and they release extracellular vesicles that are consistent with the size of exosomes. And that we profiled these extracellular vesicles, and they contain a large number, a fairly large number of microRNAs. One of the most abundant is MYR206. And what we showed, again, in vitro primarily, is that these exosomes can be taken up by fibrogenic cells, fibroblasts, FAPs, and that one of the targets of it in fibroblasts of MYR206 is RRBP1, which is a master regulator of collagen biosynthesis. And that what we think is that when satellite cells are absent, that now the fibrogenic cells overexpress collagens and other extracellular matrix proteins such that that the muscle becomes fibrotic. So we wanted to follow up on this. And so as opposed to killing the satellite cells, we more recently have wanted to label them and track them and try to understand more fully what they are doing in this hypertrophic process, because here, this communication with fibrogenic cells, we wanted to explore in more detail. And so what we did was to use the same PAX7 driver Cree mouse and cross it to a flock stop TD tomato mouse, such that satellite cells are specifically labeled with TD tomato. And here's just an image on a single fiber of a labeled satellite cell. And that this results in very efficient labeling. And here on the right side of the slide, just if you look at and count and quantify PAX7 positive cells, you can see that they are very efficiently labeled with TD tomato. So that what we did was then to do the tamoxifen treatment and a 2-week washout period, which is our standard, at least 2-week washout of the tamoxifen. And then we subjected the mice again to 2 weeks of Synergist ablation, isolated the red cells, the TD tomato positive cells by FACS, and performed single cell RNA-seq. And the results are shown here. So what we were surprised to find is that there were now, of course, red cells, but that was an interesting profile. So we were rather generous in our gates in terms of isolating the cells as shown here. And on the right side here is the actual single cell seq data. And we appended the TD tomato gene to the mouse genome so that we could actually detect it in the cells. And what you can see here is that, of course, as expected, satellite cells are the most highly labeled cell type. Some myonuclei came through the sort, and the population here, we believe, based on the gene expression profile, that they are probably derived from newly fused satellite cells. And I'll talk about that in a minute. And that we did detect fibroblasts that were red, as well as endothelial cells and some macrophages that actually expressed the transcript for TD tomato. So we wanted to explore this in more detail. And so we focused in on the endothelial cells, which at this 2-week time point was one of the most highly labeled enriched cells after satellite cells. And if you look at the transcriptional profile of these endothelial cells that either are expressing TD tomato or are not, they have very different transcriptional profiles. And one of the class of genes that was, or one of the pathways that was the most down-regulated in the endothelial cells that were expressing TD tomato were genes involved in, again, ECM organization. So we thought perhaps this was an indication that, in fact, the satellite cells may be communicating through EVs with endothelial cells, as we had shown previously with FAPs and fibroblasts. And so what we did was to actually perform another FACS, but this time also using VCAM as well as TD tomato to specifically isolate satellite cells. We did that. We put them in culture and allowed them in culture for several days, and we collected the media and isolated the extracellular vesicles from the media. And again, if you look at the size profile, that they are consistent with the size of exosomes. And what we found is that these extracellular vesicles contained a very abundant TD tomato messenger RNA. Which is we hypothesized was how these other cell types are actually acquiring the TD tomato message. And in fact, the exosomes are actually, or the EVs are actually fluorescing red, so that there's actually TD tomato protein in the EVs as well. So coming back to the original observation, and I should say that this was just recently published in iScience, and we performed a large number of controls showing that this wasn't read-through transcription or off-target recombination, that it really was the most likely explanation is that this is a transfer of TD tomato from the satellite cell EVs to other cell types. But this experiment, as I said, was after 2 weeks of overload, and you can see that there are some myonuclei that are expressing TD tomato, probably from satellite cell fusion. But then the TD tomato released and delivered to other cell types may actually be coming from the myofiber and not satellite cells. So to try to address that question, we actually just did 2 experiments with 2 different lines of mice. One is that we just did a shorter overload, a shorter term of synergist ablation, and that is at 3 to 4 days, which is prior to satellite cell fusion or prior to when we can detect any satellite cell fusion to the fibers. And the other thing we did was to generate another line of mice, TD tomato reporter mice, but which also contained the floxed NWASP gene. And NWASP is required for satellite cell fusion. So upon tamoxifen treatment, the not only do the satellite cells express TD tomato, but the NWASP gene is recombined out so that the satellite cells can't fuse. So again, I'll show you results from that mice at a very early time point where we wanted to do FACS single cell seq to see at that early time point what satellite cells might be communicating with. But our first observation surprised us because I said this is in the NWASP TDT mouse at a less than 1 week time point of overload. And what we found is that there were red fibers. And really we could detect no myonuclear accretion, we could detect no satellite cell fusion, but we did still detect TD tomato positive fibers. So we thought, well, then potentially satellite cells not only might be communicating via EVs to control ECM remodeling in a variety of cell types in muscle, but they may actually be communicating with the fiber prior to fusion. So we wanted to explore that in more detail. So what we did was to isolate, as the EVs, these from the TD tomato mouse. We isolated the EVs from the MPCs, the myogenic progenitor cells. And then we added those to just wild type myotubes. So these are myotubes just isolated from wild type black 6 mice, so no TD tomato. And what you can see is those animals that got TD tomato EVs from TD tomato positive myoblasts or myogenic progenitor cells do become red. And that's shown here in this bottom panel. And the next slide is just a higher magnification so that you can see that these myotubes do take up the EVs and they actually begin to express TD tomato. So we think that satellite cells are in fact communicating with fibers very early during hypertrophy, which makes sense because in this synergist ablation overload, there are the satellite cells increase dramatically, 300, 400%, and much beyond those that are needed for fusion. So the fact that they really have functions independent of fusion perhaps is not surprising. But again, we wanted to know what this communication might be doing. And so what we did was just to isolate the myotubes that received EVs and compared the RNA-seq profile to those that did not. And the gene that was the most highly differentially expressed in the myotubes that received EVs was MMP9. And you can see that here that the expression of MMP9 or the abundance of MMP9 message in these myotubes was decreased following being plated with MPC EVs. And just looking and profiling the EVs, as I said, that there are a number of microRNAs contained within the EVs that do target MMP9, as well as other extracellular matrix encoding genes and remodeling genes. So it really does appear that that perhaps this early communication prior to fusion may be really facilitating the eventual hypertrophy, and that the fibers too are expressing genes that can help to remodel the ECM to facilitate growth. And so here is just a cartoon of what we're thinking now is that satellite cells really do communicate and have a large role in muscle adaptation and remodeling to a variety of cell types. And these lists are not by any means complete. And what we earlier showed that they communicate with fibroblasts, FAPs, fibrogenic cells. I just showed you evidence that it appears they can communicate with endothelial cells and even with the muscle fiber itself.
4 minutes left.
Okay, so just a couple slides. I just want to shift gears slightly because I want to address the aging issue, and that is we published several years ago that when you in our reverting back to our PAC7 DTA model, if you ablate satellite cells and leave the animals just to live out their life without satellite cells, it does not lead to sarcopenia, doesn't exacerbate sarcopenia. But we wanted to see but those were sedentary mice, and so we wanted to see if we did the same experiment, but in active mice, would the depletion of satellite cells have an effect on muscle size or sarcopenia? So it's the design of the experiment, very simple: deplete satellite cells at 5 months, give that mice 2 months washout, and then we gave them access to a wheel. And what we found is that if you then look now, since they're running, we can isolate both the plantaris and the soleus muscle. And what you can see is that in those animals which only received vehicle, you see an increase both in the soleus and the plantaris and satellite cell abundance, but the satellite cells remain very effectively depleted throughout the life of these animals. And as a result, what we found is that the animals that did run and still had their full complement of satellite cells, that all 3 fiber types 2A, 2X, and 2B increased in response to running, and this was blunted in the absence of satellite cells, and similarly in both type 1 and 2A fibers in the soleus. Overall, there was a shift in fiber type in this plantaris from 2B, 2X to 2As, and since those are smaller, the mean fiber CSA was not affected, but it was in the soleus. So in conclusion, what I've showed you is that satellite cells influence the extracellular matrix in muscle through EV-mediated delivery of microRNAs to other cell types, and that this may facilitate muscle remodeling. Because in the absence of satellite cells, the muscle becomes fibrotic, which may inhibit long-term growth and adaptability. And that we believe that the cross-communication between satellite cells and many other cell types in muscle may regulate the muscle environment. And it's a word of caution in terms of equating red fibers or the fact that the reporter messenger RNA and potentially protein can be transferred from satellite cells to other cell types makes lineage tracing really problematic with this tool. I just showed you here at the end that as satellite cells are required for increased muscle size in response to lifelong physical activity, and therefore the loss of satellite cells with age may limit adaptations to physical activity, although it does not cause sarcopenia, even though it does impair regeneration, of course. And finally, I think the take-home message here is that activated satellite cells or myogenic progenitor cells play a larger role in adult muscle adaptation to exercise than simply providing nuclei to the growing muscle fiber. And I'll close with just thanking the people in the lab who've done all the work. The top list are current lab members, and the bottom list here are those who were involved in some aspects of the experiments but have moved on to other labs or running their own labs, my long-term collaborators at the University of Kentucky, and my funding agencies. Thank you.
Thank you, Charlotte. Let's see if there is any question.
Yeah, I have a question. I don't have any video. I'm so sorry. It's Rick. I put my little hand up.
Hi, Rick.
Hi, Charlotte. It's okay. Zippy, can I ask the question?
Of course.
Okay. Charlotte, I really love that stuff. I'm wondering if you're thinking that this general EV-mediated communication amongst mononuclear cells in the ECM is a one-way street, and satellite cells are the master talker, or is that just experimentally that's all you have access to, so you can only think in terms of satellite cells currently?
Yeah, no, I think it's both way. I think cells are communicating with each other all the time. I mean, there's some work from other labs. I mean, certainly, and I think Gabby has been, who is here on the call, has shown that the FAPs communicate with satellite cells, and so I think it's there. Everybody's talking to everybody.
Thank you.
So,
Hey, I asked a short question, Zipporah.
sure.
When I asked Zipporah to organize this wonderful day, I asked her, do you know someone working on a human myopathy dependent from a gene mutation related to satellite cells? You clearly demonstrated that if you inhibit satellite cells, this is very, very, very dramatically pathological. there is no one known genetic disease dependent from some spontaneous genetic defects of the genome regulating satellite cells?
Well, the one that comes to mind, although it's not necessarily clear, you know, the rhabdomyosarcomas, where you have the translocation, alveolar rhabdomyosarcomas, where you have the translocation of the PAX7 or PAX3 gene with forkhead. And although it's not clear necessarily that absolutely that those tumors derive from satellite cells, but that's sort of the thought in the field that translocation, that mutation.
Could you please send me a list of this information?
I can send you, yeah, a list, and maybe somebody else, David Goldhammer, somebody else may be able to
Because
talk
I looked
about.
to PubMed, but I was unable to find nothing related. I'm sorry, it is more than enough. Thank you very much.
Okay.
I know that we passed the time, but I think that the comment that I wish to make is very related to the point. So, Charlotte, the question that came before from Rick was, is it a back and forth highway or one way? And you said, no, it's maybe a global, just that you have the tool for PAX7. And I was wondering if somebody else is using, you know, there are tools with endothelial cells, but I would like to draw everybody's attention to a paper that was quite popular. The whole group was very popular in 1981. It was out of Connecticut, maybe from Stores, maybe David can comment, but it was maybe before his time there. And there was a very big leader. He was able to isolate myosin polyzones, Stuart Haywood. And they published in PNAS in 1981 paper with the following title: Cytoplasmic Utilization of Liposome Encapsulated Myosin Heavy Chain Messenger Ribonucleoprotein Particles During Muscle Cell Differentiation. What they did, they put it on the culture. Nobody
But these are manipulation of a normal muscle, like the
these
Charlotte,
are not manipulations. This could be actually
a disease, a myopathy with a clear name,
I'm not,
like Duchenne muscular dystrophy, in which there is a defect in a original defect in satellite cell.
Hugo, please excuse me, but I'm actually not addressing your question. I'm going back to the general team of Charlotte. I know that your question
I'm sorry, I waste enough time, but the answer is that no one is able to provide the name of this disease. Is this true or I am wrong?
I'm not talking about a disease. I'm just bringing up a very old reference that deserve attention, that in 1981.
But there is something in any medical book in which there is the name of a disease which pathogenesis is a original disease of the satellite cells.
Carsten Bonneman has a family, and he hasn't published it yet. Carsten Bonneman has a family where there's a defect in PAX7 in the satellite cells, and there's also a German family.
Why it is not this
He hasn't
popular?
published it yet. I
Why
don't know.
it is not this popular? Is it so rare?
It's
a rare disease?
Extremely rare because the patients have virtually no muscle.
Sorry, I am satisfied to astonish you, all of you, once again.